Bismillahirrahmanirrahim. Assalamu alaikum students. Today we will discuss the life cycle of Ephedra. This is the diagrammatic life cycle of Ephedra. As we know that Ephedra plants are dioecious, in which the male and female cones are produced on the different plants. This is the male cone and this is the female cone. In the male cone, microsporangia are produced on the microsporangiate shoots in the axils of the bracts, and in these microsporangiate shoots 8 to 12 microsporangia are produced at the tips of this microsporangiate shoot, in which the microspore mother cells are produced. And in the female cone, most of the bracts are sterile and only upper two bracts they bear ovule in their axils. And the structure of the ovule you have studied in detail. This is the sporophytic phase, diploid phase of the plant. In the microsporangium, the microspore mother cells are produced, and in these microspore mother cells meiosis, reduction division occurs, and haploid microspores are produced. And these microspores, when they are present inside the microsporangium, they start to germinate, and during their germination, these microspores, when they are the 5-cell stage, then they are released from the microsporangia, and this semi-germinated microspore is then known as the pollen grain also, in which in this pollen grain, this is the tube nucleus, this is the body nucleus, and this is the stalk nucleus, and these are the two corthelial cells. At this stage, these microspores, which are known as the pollen grain also, these are released from the male cones from the microsporangia. On the other side, in the female cone, in the upper bracts, ovule is produced, and in this ovule, the structure of the ovule consists of the integuments, inside which the nucellus is present. And in this nucellus, one of the cell towards the micropylar end, it becomes different from the other cell, and it acts as the megaspore mother cells. Then reduction division, meiosis occurs in this megaspore mother cell, and the 4 haploid megaspores are produced, and out of these 4 megaspores, 3 are disintegrated and only one survives, which is known as the functional megaspore. And when the microspores in the microsporangium and megaspores in the ovule are produced, then here the gametophyte stage or haploid phase is started. This megaspore, it germinates and it produces the female gametophyte, and this female gametophyte, it then produces the Archigonia inside it towards the micropylar end, and 1 to 3 Archigonia are produced, and through the micropylar tube in this ovule, on the upper side of these Archigonia, the nucellus tissue is disintegrated, and here the pollen chamber is formed, and these pollen grains or male gametophytes, through pollination, enter through the micropylar tube and then reach to the pollen chamber, and here the further germination of these pollen grains occurs, in which the pollen tube is formed, and the body nucleus also divides to produce the 2 male gametes. This pollen tube, through the pollen chamber, it enters into the through the neck of the Archigonia, and then it releases the two male gametes inside the archigonium, and each archigonium, it consists of upper 30 to 40 celled neck region of this, and one ventral canal cell nucleus and the other egg nucleus. The two male nuclei, one male nucleus, it fuses with the egg nucleus, and the other male nucleus is reported to fuse with the ventral canal nucleus. And then after fertilization, zygote is formed after the fusion of the male and female nuclei, and this zygote after fertilization is also known as oospore also. And then when zygote is formed, then cleavage polyembryony occurs in Ephedra, means that zygotic nucleus divides to produce the 8 nuclei, and each nucleus develops the wall around itself, and then this cell is known as the proembryonal cells. These are also known as the proembryos. And all these proembryos, these start to germinate, these start to develop and into the embryos, but the lower proembryos, they cells develop more rapidly, and at the maturity only one proembryo matures into the embryo, develops into the embryo, and it is developed into the embryo, and while all the other proembryos, these are disintegrated. And each proembryo, it produces the develop into embryo, and suspensor tube is formed, and the proembryonal nucleus, it enters into this suspensor tube, and then it divides further, and it produces the at its lower side, out of all these proembryos which start to develop, all are disintegrated and only one matures into embryo, and in this embryo division occurs, and in this embryo after division two cotyledons are produced, in the axils of which the plumule is produced, and then this embryo is converted into seed after some changes, and then this seed, when it is germinated, it develops the male and female sporophytic plants again, and in this way the life cycle of Ephedra is completed. This was the general diagrammatic life cycle of Ephedra. Here in this diagram, the detailed diagrammatic life cycle of Ephedra is shown. Female cones on the female sporophyte plants, and the male cones on the male sporophyte plants. In the female cones, the lower all the lower bracts, these are sterile, and the upper two bracts, they are fertile, and they produce one ovule in their axils, and these ovules consist of the long upper micropylar tubes, mean integuments on the upper part of the ovule. These are elongated to form the long micropylar tube. Here is the diagram, internal diagram. Upper bracts, these produce one ovule in their axil. This is the structure of the ovule. These are the two integuments, and this is micropylar tube, and in the basal part of this ovule the nucellus tissue is present, and in this nucellus tissue one of the cell is differentiated to produce the megaspore mother cell. And here in the male cone also, in the lowermost bract, is sterile, while all the other bracts, which are decussately arranged, these produce the microsporangiate shoot in their axils in the bracteoles. And microsporangiate shoot, it consists of group of microsporangia at their tips, and at the lower part the stalk-like part or column is present. And in the microsporangium, microspore mother cells are produced, which are diploid, and these microspore mother cells, after reduction division, they produce the tetrads of the microspores. In these tetrads of microspores, which are haploid now, germination is started, and microspore starts to germinate to produce the male gametophyte. As we discussed earlier in the last diagrammatic life cycle, microspore germinates to produce the 5-cell stage, which is known as the spore germinated microspore. It's also known as the pollen grain, and it's also known as the male gametophyte. And in this the male gametophyte or pollen grain, which is produced inside the microsporangium, the stages, the cells which are present in this stage, 5-cell stage, are the 2 prothelial cells, one tube nucleus, and then one body cell, and the other is stalk cell. At this stage, the microspores are released from the microsporangia from the male cones. On this side, in the ovule, when megaspore mother cell is differentiated after reduction division, this megaspore, it produces the megaspore mother cell produces the 4 megaspores, and in these 4 megaspores, the upper 3 megaspores are disintegrated, while only one survives, which is known as the functional megaspore. This functional megaspore germinates to produce the female gametophyte by free nuclear division. In the previous stage, early stage, in the center vacuole is present, but in later stage this vacuole is disappeared due to the distribution of the nuclei in the center also, and the wall formation occurs, and this tissue becomes, these nuclei develop wall around themselves, and this becomes cellular, and this is the female gametophyte. And then if this female gametophyte, towards the micropylar end, 1 to 3 cells are differentiated, and they develop the Archigonia towards the micropylar end of this ovule. And here 2 Archigonia are present, and the structure of each Archigonium is like this, in which 30 to 40 celled neck region is present, and in the lower part of the Archigonium, one ventral canal nucleus is present, and the other egg nucleus is present. In the micropylar tube, the microspores, which are released from the microsporangia, which are also known as the pollen grains, and these pollen grains, these reach to the ovule, micropylar tube of the ovule, and this process is known as the pollination. In the micropylar end, micropylar tube region, pollination droplet is present, and through this droplet these microspores are sticked, and these reach into the part where the Archigonia are present. In the above region of the Archigonia, the nucellus tissue is disintegrated, and this produces the pollen chamber. This region is known as the pollen chamber. These microspores or pollen grains in semi-germinated form, through the micropylar tube they enter, and then they reach to the pollen chamber, and here the further development or germination of this pollen grain occurs, in which the pollen tube is formed, and meanwhile the body nucleus also divides to produce the two male gametes. Here we can see that in the Archigonia, this is ventral canal cell and this is the egg cell, and the two male gametes which are from this pollen tube of the pollen grain, these are released into the archigonium, and one of the male nucleus it fuses with the female egg nucleus, and the other male nucleus is reported to fuse with the ventral canal nucleus, and where the male nucleus unites with the egg nucleus, the diploid cell is formed, which is now known as the zygote, and from here the gametophyte stage is ended and the sporophyte stage is started. In this zygote, the division occurs, to which we also say that cleavage polyembryony occurs, that from this zygote usually 8 nuclei are produced, and each nucleus after formation wall around itself is known as the proembryos, and all the proembryos start to develop, but at maturity only one proembryo is developed into the embryo. And in this embryo, the suspensor tube is formed, and the nucleus, embryonal nucleus, it enters into this tube, and it starts to divide, and after division, in the lower part of the embryo, the two cotyledons are produced, and in the center, in the axils of these two cotyledons, plumule region is present, from which the shoot apex will be produced, and on the upper side of this embryo the hypocotyle region is present, through which the radical part is developed, and radical part will produce the primary root. And this embryo, after producing all these parts, when all these parts are completed, then the internal structure of the embryo is like this, and some changes occur through which the embryo is changed into seed. This is now known as the seed, because integuments are converted into the testa of the seed. These form the testa of the seed, and then this seed germinates apigally. Apigally means the seed starts to germinate when they are present above the soil, above the ground. Their germination is started, and when the germination is started, then after rupturing the testa, the plumule region, cotyledons along with the plumule region, these come out from the seed, and then hypocotyle, from the hypocotyle region the radical is produced, and radical grows downward to produce the primary root, through which the again next branches are produced. They will produce the secondary roots, and then these cotyledon leaves they are developed and are released from the seeds and germinate, and from this shoot apex plumule region, the shoot apex, soon the first leaves of the first foliage leaves of the Ephedra sporophyte plants are produced, and in this way the life cycle of the Ephedra plant is completed, and the sporophytic phase alternates with the gametophyte phase. I think you have understand well the life cycle of the Ephedra through this detailed diagram, and the description of this detailed diagrammatic life cycle is mentioned here in Spanish language, You have studied in detail, so you can translate, you can write the parts, you can name the parts of all these diagrams yourself because you have studied in detail. This is the graphic life cycle of Ephedra, in which no diagrams are shown, only the stages are mentioned through their names. The male cones and the female cones, due to superficial resemblances with the angiospermic plants, these are also known as the male inflorescences or male flowers, and female inflorescences or the female flowers. We can see here again in these diagrams, these are also known as the male inflorescences, male flowers, and these are also known as the female inflorescences or female flowers due to their resemblance with the angiospermic flowers. In the male cones, microsporangia are produced, which are also known as the anther sacs. In the female inflorescence, female cone, ovule is produced. In the ovule, megaspore mother cell is produced. In the microsporangia, microspore mother cell are produced. And after reduction division, these microspore mother cells form the microspores, not macrospores. Microspores, these are microspores which are haploid. Megaspore mother cell will produce the one functional megaspore. Megaspore on germination will produce the female gametophyte, and in the female gametophyte Archigonia are produced, and inside the Archigonia egg or oosphere is produced. Microspores on germination will produce the male gametophyte, which is also known as the pollen grain, and in the pollen grain male gametes are produced, and after pollination the male gametes fuses with the egg in Archigonia and fertilization is effected. Fertilization occurs, and after fertilization this egg is now known as the oospore, and in this oospore or zygote cleavage polyembryony occurs, means that this zygote divides to produce about 8 nuclei, and all these 8 nuclei they form the pro-embryos. After formation the wall around themselves, and all these pro-embryos they start to develop, but only one pro-embryo it reaches to maturity, and it is known as the embryo. And in embryo, when all the parts are completed, some changes occur around this ovule, and this embryo is converted into the seeds, and seed on germination will produce the Ephedra sporophyte plants again. So students, today we have discussed the diagrammatic and graphic life cycle of the Ephedra plant.